Hi, my name is Arthur Liang, and I'm a high school student studying at Saxon High School in New York City. Alcohol use disorder, or AUD for short, affects over 10 million people in the US, and is a neurological disorder marked by an addiction to alcohol that can lead to health problems such as heart disease and liver damage. Investigating how AUD affects human neurons may lead to promising insights for developing effective drugs and targeted treatments for patients. In my project, I provide a vital stepping stone for this goal by studying how neurons respond to glutamate, one of the brain's major chemical signals, and how chronic ethanol treatment affects it. I chose my project because my father struggled with alcohol dependence in the past and was able to overcome it once he had me and my brother. It was fascinating to hear his story 2 years ago about how it affected his social circle and even his job, which was essentially his lifeline as a new immigrant to the States. I'm glad to say that he's been sober for over a decade now and won't even have a sip during holiday or birthday celebrations. I admire his perseverance, but I think he's a special case. Many others aren't as lucky and continue to fall deeper into the hole. And alcohol abuse, like many other addictions, takes a lot of willpower and discipline to recover from. And so I reached out to the Schlesinger Laboratory knowing I wanted to study alcohol use disorder in a functional assay to learn more about how heavy drinking affects human neurons. I was really interested in neuronal excitability, and I felt that this research project and topic, because it's so near and dear to my heart, is very fitting for my high school capstone research project. My results also demonstrate that I now have a high throughput strategy, allowing me to gather functional information from a high number of neurons simultaneously. Using this strategy, I can further examine how neurons are affected by ethanol on a wider scale, aiding in understanding the functional consequences of ethanol on human brain circuitry, and eventually in developing therapeutics to modulate AUD and other disease states. It was important to use human stem cells instead of relying on animal models, which for this type of research are typically of mice, because stem cells overcome several limitations observed with animal models that typically focus on manipulating a single gene and thus fail to recapitulate the full scope of molecular and cellular phenotypes observed in humans. And so this way, it was quite easy to not work with an animal model, and this likely enhanced my research because I ended up working with human neurons instead of mouse neurons. I will be starting college at MIT this fall, hoping to major in brain and cognitive sciences and computer science. And yes, I am planning to continue this research. And lastly, as cheesy as it sounds, I like to emphasize the timeless adage that if there's a will, there's a way. I believe that there are always alternatives to methodologies that rely on animal experimentation, and sometimes, as in my case, they might end up being more effective. Thank you.